Hello everyone, welcome to our second lecture. So we're going to be looking at innate immunity again. So again, here's a good figure to just get a good overview of all the cells in the innate immunity, and we'll be slowly going through these in detail and some of the processes. So again, here's the figure we looked at last time. So innate immunity lacks specific memory, but trained immunity is a recent finding, so it's not completely mindless. So, as we've discussed, immune response is a continuum of events. There are ongoing interactions between innate and adaptive immunity, and innate immune responses provides protection while adaptive responses prepares, gives it time. Innate responses can influence the adaptive response, and the adaptive response can both improve and regulate the innate response. So, there are PAMPs. So, this is a term that stands for pathogen-associated molecular patterns. PAMPs are recognized by PRRs, or pattern recognition receptors, on certain types of immune cells, and we'll discuss these in detail. But first, we're going to discuss the relationships of the microbe and the host. So, host-associated microbes are considered symbionts. So, these have relationships to the host which are based on the net fitness advantage or disadvantage they provide to their host. So, microbes can be mutualist, where both the host and microbe benefit. They can be commensal, which you may have heard this term before, where the bacteria or cell benefits from the host, but the host is not affected. And we can have a pathobiont, which can be a mutualist or a pathogen, and a pathogen, which is something, considered bad, and it can damage the host. So the pathogens can damage us in different ways. So they can damage us through immune complexes, which can cause inflammation and tissue damage, often at the kidney. So usually when we have an attack, you can see here the complex, these will end up getting stuck in the kidneys when there's filtration occurring and cause damage. We can also have mistakes of cell-mediated immunity with overactivation causing autoimmunity. And we can also have induction of cross-reactive antibodies, which target our own tissues. Here you can see the different things, complexes, antibodies, and cell-mediated. Here we can see the different types of pathogens that can cause these diseases down here. So several can cause kidney disease, rheumatic fever, and you can see here meningitis and tuberculosis and more. So an example would include the potential complications caused by Campylobacter infection. So you can see here the carbohydrate structure of LPS and the carbohydrate structure of GMI, where we have cross-reactivity. So basically antibodies that are formed against this LPS can cross-react with the GMI. And this can cause Guillain-Barré syndrome, which is an autoimmune disease, which can cause paralysis due to the damage to the peripheral nerves. This is due to the cross-reactive antibodies, to the gangliosides on peripheral nerves. So we can see here antibodies against LPS of some Campylobacter strains cross-react with GM1 gangliosides in the myelin sheath of nerves. This is basically due to the structural similarity that we discussed up here, and this leads to the damage and loss of nerve conduction and to paralysis. So we can see here the common routes of infection. So most of us know that we can get infected through the mouth and respiratory tract, stuff we eat, and also through the reproductive tract, such as STIs and blood transfusions, such as like AIDS. So this is just a good figure to just get a good overview of different pathogens and their disease effects. So quick example, inhalation or ingestion, influenza, everyone should know about that. And then types of pathogens we can see here. Same thing with spores and allergies and other stuff like that. Same with gastrointestinal tract. We can have, salmonella, which is a common one. And then STIs, we can see here, hepatitis B and HIV. The impact of the route infection reflects the ability of the microbe to grow and cause damage, and at a given location and the host defense is available. So depending on where we encounter the pathogen, and how much and what pathogen it is, we can have different responses through our innate immunity. This is where that trained immunity comes in. Oops, let me go. So here we can see more routes. We have external surfaces. So, sometimes some things can actually infect us just through physical contact. So example would be athlete's foot, where we have a fungal infection. Wounds and abrasions open the skin up to infection. to things that normally aren't supposed to get in, and same thing with insects, which you've probably heard of ticks causing Lyme disease. So damaged epithelial surfaces provide routes for infection for various pathogens. This includes soil-associated bacteria, animal-associated, including zoonotic infections, and vector-borne. So, and a novel innate defense that is quite new and very interesting is the bacteriophage. And so these phages act as a host defense in the BAM model, which is the bacteriophage adherence to mucus. So these bacteriophages, you can see here, they have immunoglobulin-like domains, acting similar to immunoglobulin, as you'll see in the mucus. They bind to certain pathogens. So we can see here, our epithelial cells secrete mucus. These bacteriophages act like immunoglobulin, and they're kind of just free-floating in the mucus. And essentially, they can adhere to to microbes and form antimicrobial layers. And basically, they will pull, you can see they will clear it. So these mucus-adherent phages have increased chance of replicative success when they bind to a bacteria, and then these get moved through the mucus. So the bacteriophage is found in various body regions, even though they don't affect our own cells. Recent findings show that bacteriophage transcytosis occurs across epithelial cells, and they assume that the average human body transcytoses 3.1 times 10 to the 10th phages per day. So yeah, you can see here the bacteriophages activating a bacterial cell, and a new study shows that a sea of them may protect the human body. So this is an interesting area of research, and you guys can check out the paper here if you're interested. So again, we have many innate defenses. We have barriers, complement, and antimicrobial proteins, which include C3, defensins, and Reg3. We'll get into complement in the next lecture. Our innate immune cells, including macrophages, granulocytes, and NK cells. And then our adaptive cells, B and T cells. And like we said, novel mechanisms are still being discovered, including the bacteriophage. But there's also another example, which is the innate lymphoid cells, or ILCs, which we'll discuss right now. And again, in spite of all this, we still get infection, so it's not perfect. And again, infectious agents must overcome the innate host defenses to establish a focus of infection. So if everything was stopped here by these cells, we would never get sick. But as you can see, we're going to have to deal with it, and then we're going to transport it to our lymph nodes and deal with it. So again, infections and responses to them can be divided into a series of stages. A local response may prevent infection from progressing. If not, it still helps to contain the infection, though they're not useless if they can get past it. Delivery to local lymph nodes initiates this adaptive response. So again, as we said, location does matter. So different elements of the immune system are involved in defense at different locations. So pathogens may infect the different body compartments where they encounter different host strategies. So we have extracellular versus extracellular pathogens. So defense against extracellular pathogens often relies on the actions of cells and soluble mediators, including phagocytosis, complement, and antibodies. Whereas intracellular pathogens, defense against intracellular pathogens involves cells of innate and adaptive immunity, such as NK cells and CDLs, or cytotoxic T lymphocytes. And also macrophage activation is also an important feature. So we can see here the different, essentially what I've just said. So we got an extracellular, then we got a complement and antimicrobial peptides. And intracellular, in the cytoplasm, we have NK cells and cytotoxic T cells, and our T and NK cells. So key factors of innate immunity, as we discussed before, there's the phagocytes, which include neutrophils, monocytes, and macrophages, which are important in antibacterial defense. We have our NK cells, which are very important in antiviral immunity, and also have immunosurveillance role against tumors. And there's also other types of innate lymphoid cells called the ILCs. So the ILCs are a recently discovered cell type with some features of lymphocytes. A key difference is that ILCs do not have highly specific antigen receptors that characterize T and B cells. There are 3 groups, 1, 2, and 3. NK cells are now in group 1, which is important to remember. And there's a video here on, if you search this on YouTube, innate lymphoid cells from Nature, really great video. So again, we can see here, these are now in the same group, but you can see they both release IL-12. They're induced by IL-12, and they release interferon gamma, perforin, and granzyme, and are effective against viruses and intracellular pathogens. Where we have ILC2, which is induced by IL-25, 33, and TSLP, and it releases IL-5 and IL-13, and is involved in parasites. It's important to keep in mind these cytokines, as we'll discuss, how they relate to T helper cells and how these different kind of pathways work in specific kind of pathogens and responses. And then we also have ILC3 or LTI, which lymphotoxin cells. We have IL-20 induced by IL-23 and released 22 and 17 and involved in extracellular bacteria and fungi. So group 1s make interferon gamma, which promotes defense against intracellular pathogens, same as Th1 as I was saying earlier. The group 1s include ILC1 and NK cells. Group 2s make IL-4 or interleukin 4 and promote defense against parasites, similar to Th2 cells. And the group 3s make IL-17, which promote defense against extracellular pathogens, similar to Th17 cells. As I was saying, ILC groups are based on similarities to certain types of T helper cell subsets, and these are the ones we just circled here, and are defined by their cytokine production profiles, cell surface markers, and transcription factor expression. So we can see here, these are the signature stimulating cytokines and then the producing cytokines. As you can see, they can have very distinct pathways and very different that allows them to be distinguished from one another. The roles of ILC are varied but are still being defined, so the evidence on this is not completely concrete, but more is being learned every day. So ILC's main roles are at the mucosal and epithelial surfaces. ILCs play roles in the gastrointestinal defense and disorders. For example, IL-3 is involved in defense against certain gastrointestinal pathogens, but may be involved in inducing a tolerance to the gut microbiota. But ILC3s are implicated in inflammation and damage in Crohn's disease, which is a type of inflammatory bowel disease. For another example of this balance is ILC2. So they also play important roles in various airway disorders. So ILC2s are implicated in asthma, as are Th2 cells, but this highly relates, as you'll see later, this kind of relates to the cytokines they release. But ILC2s are also involved in airway repair after influenza infection. So we can't just say one cytokine is good or bad. It all basically depends. So this is an amazing figure from this paper that, again, I recommend you check out. It's kind of a yin-yang symbol of the control of inflammation and the inflammatory diseases that ILCs can cause. So in the blue, we have all the, good effects. So we can see ILC3, 1, and 2. So as we discussed, we can have repair, but also can cause chronic sinusitis, asthma, lung fibrosis. ILC1 can help with control of digestive intracellular pathogens, but it can be involved in Crohn's disease. And then ILC3, like we said, can help with skin repair and wound repair, but it's also involved in other things such as psoriasis and obesity and airway inflammation. So again, as we said, neutrophils, monocytes, and macrophages are the key phagocytes and have important antibacterial defense mechanisms. So we've looked at this figure before, but phagocyte products that kill microbes are below. So these are all the different things they release. Several bactericidal agents are made or released by phagocytes after microbial uptake. Some act in the phagolysosome, and some are secreted and can damage the host. So there can be some friendly fire per se. So we've looked at this figure before with the release of the phagolysosome and all of its contents. So we have the production of our reactive oxygen intermediates, also sometimes called the reactive oxygen species, including hydroxyl radicals, superoxide anion, and hydrogen peroxide. We also have inducible nitric oxide synthase, which is activated to result in nitric oxide production. We have increased production of lactoferrin, defensins, and destructive enzymes like lysozyme proteases. But we also have neutrophil extracellular traps, or nets. So these nets, neutrophil nets, form as neutrophils die, and these are fibers of DNA, histones, and proteins that trap bacteria and kill with antimicrobial peptides using defensins. So here we have the normal process I was talking about before, where we have NADPH help with releasing the reactive oxygen species. We have the neutrophil keep killing, and then as it dies, it releases the net, or extracellular traps. Here we can see the two types of figures, and these will kill bacteria as well. But some pathogens can manipulate this host defense against us. So for example, we have Staphylococcus aureus, which can use the neutrophil extracellular trap components to induce macrophage death. So basically, they can protect the bacteria. So as the nucleus breaks down DNA in the nets, S. aureus uses adenosine synthase A to generate dioxyadenosine, So deoxyadenosine activates macrophage caspases, which promotes pro-caspase 3 conversion to caspase 3, and this induces macrophage death. Caspase 3 is also involved in some other important mechanisms of actually killing things we don't want. But just keep that in mind. Caspase 3 is usually involved in something getting killed. S. aureus also damages APCs and lymphocyte activity by producing adenosine. So again, how respiratory burst is not, it's very important, and it can be involved in certain conditions. So this is an image from Cells Alive, so you can tick this link if you want. It's an image of the respiratory burst using the indicator dye nitroblue tetrazolium, or NBT. This image shows a neutrophil killing a yeast cell in a time-lapse video, and is color enhanced to show the degree and location of the respiratory burst. So this figure is kind of just where you can see, oh, if this goes wrong here, or here, this is where issues can occur, but we'll go into more detail later. So anyways, we're at the end. So in our next lecture, we're going to be looking at pattern recognition receptors, which is the mechanisms of recognition by phagocytes, so PRRs, which you mentioned before. So PRRs recognize the PAMPs, or the pathogen-associated molecular patterns we discussed, and also MAMPs, which are microbe-associated patterns. There are numerous types and categories of PRRs. There's phagocytic receptors, secreted receptors, singular receptors, and transducing receptors, both membrane-bound and intracellular. And so here we can just see a good graph, a good table of the comparison of characteristics between recognition molecules of innate versus adaptive immune system. So again, simply specific inherited, yes, this is not inherited. So this is, I'm not going to go through this whole thing, but this is something we'll go through, worth going through, just to give yourself a good starting point. So anyways, that's the end of the lecture. Take care and see you in the next one.